Hey guys, welcome back. Uh, just double check if everything good. All right. So this is the urinary system, and we are going to move on to the final portion. I hope that's the final portion. All right, let's see. So we talk about everything about the kidney, and we talk about how the um the urine transport from the kidney to ureter to the bladder and then to the urethra. So urination, that behavior, we also talk about those. We also talk about how does the bladder, full bladder, will trigger the parasympathetic nerve to contract the bladder, and also will tell your consciousness to decide whether you're gonna pee or not. And so the urination or micturition is right here. This is a good summary. Urination or micturiation that is controlled by both autonomic, the parasympathetic nerve to contract the bladder, as well as your voluntary control to relax your external urethral sphincter. So you got to have both so you can urine. Knowing all this, the next thing we look into is the how does kidney produce this urine, and we look into the functional unit of the kidney, which is nephron. So we look into the nephron and we see the interaction of the blood vessel and the nephron, and we focus on two blood vessel, which is capillary, which are capillaries. Two groups of capillary. One is one is glomerulus, the other one is peritubular capillary. So it's right here, glomerulus and the peritubular capillary. They serve different function. Capillary is the place we have substance exchange. So we have different type of substance exchange in glomerulus as well as in the peritubular capillary. So these capillary are working side by side with the nephron. So we use the nephron uh, as a guide to see what's going on through these interactions. So we will start by looking at the Bowman's capsule, then uh, proximal, loop ventral, distal, collecting tubular, collecting ducts. And so that's the journey we are going through. Basically, it's right here. This is a picture here, Bowman's capsule, proximal, loop ventral, and so this is several turns here, and we will go through it. Each turn has its feature. Yeah, so it's very unique. And basically it's good summary. Life can be complicated, but this nephron can be quite clear and also easy to understand. So here is just some labeling that you need to know, and so you need to be able to label. Right. So we talk about the beginning, Bowman's capsule. The function is filtration. How about the next one? How about the remaining one? So we are going to go through the Bowman's capsule, and this lecture, I hope we can finish all this. Proximal, loop handy, distal, collecting tubule, collecting ducts. All right. So let's start by looking at where were we? I think we were here. Yeah. So this is proximal. So what does proximal do? You need to know that proximal is the first segment right after Bowman's capsule. And you need to kind of remember that what does Bowman's capsule do? Bowman's capsule is a place that we start to receive urine. And how does Bowman's capsule receive urine? It's all by force because here, let's go back to here. Here, the glomerulus, this is the blood, this is the capillary. Glomerulus got pushed. The blood coming here got pushed. So they got squeezed. They got pushed over into the Bowman's capsule. So that's how the Bowman's capsule collect the urine at the first place. And so this, you need to know that everybody is get forced got pushed through. Do you like to get pushed? Do you like to um do you like to be pushed to do things? You don't like it, right? Nobody like to be pushed. And it's something here. So when a lot of stuff get pushed to go through coming into the Bowman's capsule, they don't like it. We don't like it. Nobody likes it. But it's the first. It's a must. Even though uh it's not what we want, but that is a must because then we can do the remaining. So that's because when everybody got pushed through, why do we why it's not good? It's because that when a lot of things got pushed through, a lot of good stuff also got pushed through. So I show you here that this one here. So a lot of good stuff like glucose. Glucose got pushed through. Glucose is small, so he has no choice. He got pushed through. Amino acid. Amino acids. It's good stuff. You have to eat, digest. Then you can um produce this glucose, absorb it in in your body, and you don't want them to get urinated, but well, they are small, so they got pushed through, so they got urinated. And so that is the situation. And that is what we are going to face in the proximal. So because a lot of good stuff got pushed through, and so the very first thing that we want to do is to take them back before we lose it, right? We don't want to lose it. So before we take them back. And that's why in this beginning of the nephron, it was kind of designed in a way that, well, the tube is not straight. If it's straight, everyone got pushed through like your toilet, right? Everything got pushed through so quickly that you have no chance to take them back. But here it's not. They have multiple turns. So kind of a beautiful design that it make it difficult to flow through. Even though everybody got pushed through, nobody got flow through that easy. So let me show you the exact one, the real one right here. Not here, but I like this one better. So this one you can see that that's why we call it convoluted, proximal convoluted tubule. So it has multiple turns. Yeah, so you know, well, you got pushed through, but you are not going to flow away. And so using that environment, what we do over there is take it back. So the main theme, each segment has its own theme. The first one is filtration. Bowman's capsule is filtration. Proximal convoluted. What is the main job here? The main job is reabsorption here. So the main job here is reabsorption. So you can see that just by the picture, this picture basically summarizes everything. We reabsorb glucose, we reabsorb amino acid, we reabsorb water, we reabsorb salt, we reabsorb ions, basically. Yeah, so those are what we reabsorb. We basically reabsorb all the good stuff in the proximal convoluted tubule. So we have no regret. We push everything through. A lot of good stuff also get pushed through. A lot of bad stuff get pushed through, which we want to keep them here, so we will urinate it out. But some good stuff, we also push them through because they are small. And so we don't want to lose them. And so proximal reabsorb them back. We also secrete something into the urine. The main thing got secreted into is hydrogen, acidic, H plus basically. Because our body tend to produce acidic environment, tend to increase the acidic level. So some H plus will get secreted into the urine, so we pee them out. So in the lab, we're gonna examine the urinary pH level, and so you should know that our urinary tend to be a little bit acidic. All right, so let's look at the In order to know what's going on here, let's look at this tube. So this tube basically is single layer of epithelial cell. So here is epithelial cell. Epithelial is skin. So skin coming in and in. So that's all epithelial. All digestive tracts are all epithelial. And the urinary tracts are all as epithelial. Yeah. So the inner layer is the epithelial. So these epithelial cells has two sides. One is internal is lumen, so or we call it luminal side or apical side. The outer portion is the basolateral side. So just some orientation, so you know, as we talked, mentioned or described in the very first lecture, every cell has sodium, potassium, ATPase. Every cell has that pump. And so every cell has a negative membrane potential, and every cell has high potassium inside, low sodium outside. So every cell has it. Now we also have sodium potassium pump in that epithelial cell. Same thing, just like every other cells. However, the location is critical though. It's because that when you have this sodium potassium pump, let me use drawing first. Let me get a pen and notation. All right, good. So when you have this sodium potassium pump, you're gonna pump sodium out, potassium in. If you have the pump right here in the apical side, you're gonna pump sodium out. You're gonna pump sodium out. And if that's the case, you're gonna lose sodium because sodium gonna flow into the urine and you're gonna pee them out. No, no, no. Sodium is also very critical to our body. You have to buy salt, in the in the uh in the older time, when we cannot get the buying salt, only people living by the uh, ocean or seawater, they can get the salt, and the salt become very expensive at that time. Now we can transport, we have very developed society, so everything got exchanged very easily. But during the older time, it's very difficult to get salt. So salt is very expensive, and we don't want to lose salt that easily. And so you need to know that critical. You need to know that this sodium potassium pump is located on the basolateral side, so we can pump potassium in while pump sodium out. When we pump sodium out, we don't lose them. We store them. We still keep them in our interstitial, keep it in our body. So you need to know that. So here is the sodium potassium ATPase. They are located in the basolateral side. Yeah. All right. So that's that. So you have that now in mind, and we can start to see how do we reabsorb glucose, amino acid, and some other things. Yeah. This membrane is also called brush border membrane. Um, so you can see that it's easier for, you need to know that lumen is not that big. It's with, I don't have the real histology here. It's much smaller. So fluids go through, they will get easily contact with this brush border membrane. And so things can get transport back. Yeah, so let's see how do we reabsorb. So terminology here is reabsorption. It's because that these are originally in your body. So it's already absorbed, but you pee them out, so you kind of reabsorb them back. So this is reabsorption. That's the main theme in the proximal convoluted tubule. All right, so how do we reabsorb them? First, let's look at the sodium reabsorption. That one is easy. When you have the sodium potassium pump here, sodium pump back to our body, so sodium is back. And so basically the situation here is that you have this, where is that here? Sodium potassium in the basolateral side. they pump the sodium back to our body, sodium goes to the blood, reabsorb them back. So you create a low sodium environment in the cell, and so sodium can come in back, and the cell get transport. So you create that low sodium in the cell, and so by diffusion, by concentration gradient, sodium will move from high concentration in the urine because they got, pushed into the urine, into the low concentration. There is basically very little sodium inside the cell. So sodium got pushed back. Water will get reabsorbed because from high concentration to low concentration, urine is very watery. So water will get diffused across. It will also go through the intercellular space. Water will get absorbed. Yeah. So that's how we reabsorb sodium. We reabsorb water. When we reabsorb sodium, water follow that concentration is osmolarity. And so everything, so these two got reabsorbed. We also reabsorb glucose, amino acids, and some other ions, and some other vitamin. So their reabsorption use a co-transporter, which will be sodium co-transporter. So when sodium go diffuse over by concentration gradient, through this transporter, they will carry glucose together with them. They will carry amino acid with them. So by doing so, you reabsorb basically everything else. You reabsorb sodium, you reabsorb water, you also reabsorb glucose. We actually reabsorb 100% glucose in the proximal convoluted tubule. We suppose not peeing out glucose at all, zero. It's very valuable stuff for us, for human. For mammals, basically, is the primary source of fuel. Amino acids is important building block for protein. And so vitamin is also very important. So we reabsorb them back by sodium co-transporter. I might have that quiz question, I'm not so sure, but a quiz question can be that all this transport, all this reabsorption is by a co-transporter with which ion? And the answer will be with the sodium. So when sodium is abundant, they move, they got reabsorbed. And while they move, they also carry, using this unique transporter, when they move, they also carry glucose with them. And a different one though, they also carry amino acid. And a different one, it will carry the vitamin with them. So that's that. So by doing so, you reabsorb those good stuff. And the other, we don't really care. They will just pee them out. We will just pee them out. We also reabsorb bicarbonate. Yes. So bicarbonate in the urine, here is the picture showing you or carbonic acid in a way. We take them back. Yes. So bicarbonate is important buffer in our body. So we need to keep them. It's important buffer media in our body. And so in the blood, we have this big amount. They will get pushed through, like everyone else. But very quickly, in the proximal, we reabsorb 85%, then do plenty, reabsorbed remaining. So we basically reabsorb almost like 99% of the bicarbonate. How do we reabsorb them? The way we reabsorb them is by secrete hydrogen. So here is the bicarbonate. Let me show you here. I think I have, yes, I don't. So no, I really didn't get too detailed about it. But what you need to know here is that two things. Let me write down here. So two things. In order to, here is the bicarbonate got filtered through, in order to reabsorb them back, our body secretes hydrogen. When our body secretes hydrogen, then we can convert the bicarbonate back to the the uh, the these products. Then we can reabsorb them back. By doing so, we secrete hydrogen. Yeah. Yeah. So there are some more process into it, but because I don't want to get it too complicated for you guys, and so two things I will hope I just you can you the two things you need to kind of memorize it is that we reabsorb bicarbonate because it's important buffer media. At the same time, we secrete hydrogen, and so your urine is acidic. Yeah. So that's that. All right. So this one summarize everything we just talked about. Let's see that. We reabsorb a lot of good stuff. Amino acid and glucose, we reabsorb them 100%. We also reabsorb about 2/3 of the sodium. By absorbing 2/3 of sodium, we also reabsorb about 2/3 of the water. So water follow the concentration gradient, follow the osmolarity. So when you reabsorb the solute, water will follow it. We also reabsorb 85% of the bicarbonate. So that's what we absorb. All right, so that's the main thing. In the proximal convoluted tubule, the main job is reabsorb those good stuff back to our body. Yeah. All right, so that's that. All right, so the next stop will be loop of Henle right here. So that's our next stop. Now we go through the first one. We know what is a feature over there. We go through the Bowman's capsule. We go through the proximal. What's the next one? What does this one do? Why do we need to have this one? This one is unique because they deep down into the kidney. As you probably know that the kidney structure has cortical region and the medulla. So this is the gross anatomy portion. So, but I can remind you that here you go. So you have the cortical region, cortex, and the medulla. You need to know that this separation is not just because of they are upper, they are lower. It's because that the interstitial environment is very different. Medulla is very salty area. So this environment is very salty. This one has high, very condensed. This outer portion is normal, just like your interstitial, but this portion, we don't really have good circulation in the medulla. So medulla, it remain to be very salty. Yeah, we do have blood flow, but not as profound or not as much as the cortex. So here is the interstitial remain very salty, very condensed. So that's that. And you may ask, okay, we know that environment, we know that's the situation, but what caused it, right? What is the driving force to cause it so condensed? Is the loop of Henle. So this loop of Henle serves that function. The loop of Henle, here is the Bowman's capsule, proximal, then loop of Henle, loop of Henle, loop of Henle coming down here. Then here is the distal, then collecting tubule, collecting ducts. This loop of Henle is unique in a way that half of these loop handy is water impermeable. So in the proximal, it's water permeable, so water get reabsorbed back. And so water remain permeable, but that permeability gradually drop. This tube become more and more sealed. So water cannot go through that easily, gradually. It drop to a level that in this turn, we call this one uh, descending right here, descending limb. So this is descending limb. Descending limb, still water permeable, even though it's gradually drop. But the ascending limb right here, ascending limb is totally water impermeable. So you can have some water, you have a lot of water got reabsorbed. You gradually lose that reabsorption. So this one, so we don't dilute out this environment. So, but in the end here, is this water impermeable? So that's one thing. Water become less permeable and become totally impermeable in the ascending limb of loop handy. So that's one thing. The second thing is that on top of it, these loop of handy seek reabsorb about a quarter of sodium back to our body. So you can think that water didn't get reabsorbed, but you reabsorb about a lot of sodium in it. That's how you add salt to it. That's how you make this environment so salty. And so that is a feature. So let's go back to here. So what's the feature about the loop of Henle? Loop of Henle right here. Loop of Henle in the descending limb, you still have some water permeability. Water got reabsorbed, but gradually you lose it. And down here, you still have some, but you lose it. And in the same time, you reabsorb salt. So you make this environment less watery, but very salty. In the ascending limb is totally water impermeable. Basically, from here on, it's all water impermeable. You cannot take water back. You see those are not just the past um, uh, simple diffusion. This one require a water channel. So this is not like the before that is leakage. It's because here you add water channel to it. So we will talk about those later. But basically these, from these points on, this epithelial tube become very sealed. So water cannot go through. However, here, particularly in this deep region, you reabsorb a lot of salt. So you make this environment very salty. So that's a feature about the loop of Henle. I think I summarized here. So that's the one slide only. You need to know that the descending limb reabsorb about 20% of the water. So earlier we reabsorb about 65%. Here we reabsorb about 20% of the water. But we reabsorb about 25% of the sodium. So it makes it very salty. We also reabsorb about 10% bicarbonate. So a lot of ions, compounds got reabsorbed. It makes this environment very condensed. So that's that. That's loop of Henle. That's its function, to make kidney medullary area or kidney medulla very salty. Yeah. All right. So next up is coming back. So they leave. So it's like journey. It's like hiking trail. We start to going in. Make a lot of turn, and then going into very salty, very dark, very condensed region. And then now we come back. We can see the lights again. All right. Wow, it's so different. Why is that different? What is the unique about these distal portion? All right. Why do we need to come here? Why do we need to have it? Why is it important? The importance about the distal portion is that even though it's called distal, it actually nearby the entrance. Let me show you the exact picture right here, right here, right here. So you can see that this is proximal loop band distal. Even though it's distal, it's coming back. It's coming back into the entrance area. You can see another one here. You have the Bowman's capsule, proximal loop band, and then coming back distal. Distal is nearby the entrance area. So this is the unique part about distal. It's very much like it is journey, but this route coming back to nearby the entrance area. This unique design right here, this unique design right here has special function. It's because that your afferent arteriole, your efferent arteriole glomerulus kind of go through it. It cross through it. You can see this one even better. Afferent arteriole coming in, then efferent arteriole coming out. They they uh they um they pass through the distal distal convoluted tubule. So they pass through it. And so this distal convoluted tubule is important to send signal to the entrance. This distal convoluted tubule, we call it macula densa. So right here, let me show you here. Right here. So this is the distal. This distal, we call it macula densa. So as I showed before, the afferent coming in to the glomerulus and the efferent coming out, they all pass through the distal. This distal has very unique cells. This distal has unique cells. This distal is important when this distal right here. Let me draw it here. When this distal here, we secrete sodium. So it's sodium, sodium, sodium. But we take sodium back. We take 65% sodium back. We take 65% of sodium back in the proximal convoluted tubule. We take about 20% sodium back. We take sodium back. But still, we have a 65, 20, so that's 85. We still have 15% remain. And that 50% is important because if today is a good day, we have a lot of sodium go through. So say you have 1,000 sodium. And so even though you still have 15% here, it's good amount. And so the macula densa will say that, okay, we have good day. Nothing's going on. Just keep on doing it. So you don't need to change anything. Nobody need to change anything. We are doing the clearing. And so blood is happy, body is healthy, nothing need to change. So that's one situation. However, if the situation is that you only have, say, it used to be 1,000, but now it's only 100, right? Much less. And after the reabsorption, you remain much, much less sodium in the nephron. And that low sodium inside of the uh, nephron. This low sodium will get sensed by macula densa. And then the macula densa will need to do something because, man, today we just don't have a lot going on here. And we got to increase filtration. We got to push more into it because we don't have a lot here. And that's the job of the macula densa. And the macula densa is nearby the entrance. So they can tell our body to improve the filtration. How do we improve filtration? Remember, filtration is driven by filtration pressure. How do we increase the filtration pressure? We basically increase blood pressure. So this is another way our body regulates blood pressure. When the blood pressure is low, we have less sodium to get pushed through. We have less filtration. We have less sodium to get pushed through. We have low sodium in the distal convoluted tubule. When the sodium level is low, macula densa will tell the kidney, will tell these cells, juxtaglomerular, sorry, juxtaglomerular cells to tell them to release renin. Wait a minute, are we seeing renin again? Yes. Yeah, so this is how we see the renin, how we connect this one with our earlier cardiovascular system. We talk about the blood pressure, we talk about very important hormonal control. Blood pressure is controlled by two pathways. One is autonomic, that parasympathetic, sympathetic affect the cardiovascular system. So that's one pathway. The other one is hormonal control. Hormonal control is RA, RAAS, RAS pathway, renin, angiotensin, aldosterone, or ADH. So that's right here. So this is the hormonal control pathway. That when sodium level is low here, it will tell the juxtaglomerular cells to release renin. Renin will convert angiotensinogen to angiotensin 1, and angiotensin 1 to get converted into angiotensin 2. Angiotensin 2 acts on vasoconstriction. Angiotensin 2 acts on three locations: vasoconstriction, adrenal cortex, which is to cause aldosterone secretion, posterior pituitary again, which is to cause it to release ADH. So that's that. So that is a function of the distal, because and the particular name of that distal is macula densa, right here. So that's that. All right. So we finish the distal. The next step is collecting tubule, collecting ducts. So what's that important? Why do we need to care about the collecting tubule? Let me close this one. Clear all. The next one is, so here's picture, summarize what we just talked about, renin angiotensin 2 pathway. So here is kidney. It produced renin earlier with the kidney, but now we know where kidney where is the juxtaglomerular cells that produces. You need to know that the thing can trigger it to release, including the sympathetic, including the low blood pressure, including the low sodium delivered to distal tubule. So that's the one we talk about right here. And that will trigger the kidney to release renin. And so this pathway, we probably have the same quiz question again, or the same exam question again. Yeah. So, but lucky you already know it. You're just connecting back to the earlier lecture. Just another picture, but you already know, right? Low sodium, dehydration, sodium deficiency, or hemorrhage. It will cause it will decrease the blood volume, and then liver release produce angiotensinogen. This one decrease in blood pressure and the low sodium. It will cause kidney juxtaglomerular cells to release renin. Renin will convert angiotensinogen to angiotensin 1. Angiotensin 1 circulating in the blood. When they circulate in the pulmonary circulation, the enzyme ACE will convert angiotensin 1 to angiotensin 2. Angiotensin 2 acts on three locations. One is the vessel, one is the adrenal cortex. It doesn't show it here, but you should know that it also acts on the posterior pituitary again to release ADH. Yeah. When it acts on vessel, cause vasoconstriction. When it acts on the adrenal cortex, that will release aldosterone, causing the potassium reabsorption and sodium, potassium, yes. It basically regulate the sodium potassium level. Okay, let me correct that. So this one is not exact, right? But it regulates the sodium potassium level. And because that, oh, increased potassium can also do that, yes. So it will release the aldosterone. Yeah, it increased sodium reabsorption and potassium secretion. That is what's supposed to be. So that's that. Yeah, I think that's it. Yes, so that's that. So let's move on to the distal. So now, it's the final stop. And each one, each segment has its feature. And so it's very easy. It's very clear. It's very easy to understand. Uh, don't thank me. You need to thank nature. That's how it goes. Yeah. So what is the feature about the distal? So the feature about the distal is that this is where hormones act on. We have two hormones act on the nephron, the aldosterone and the ADH. So this is where the hormones act on. Yeah, so that's it. So let me see here. Aldosterone. Aldosterone will act on the distal collecting tubule collecting ducts. Aldosterone is a mineral corticoid. So they will act on the mineral corticoid receptor. What does it do is that it will act on the gene expression, the nucleus, there's DNA. So they will act on that DNA to cause these cells to produce more ion channel. We have the sodium channel, we have the potassium channel, we have the sodium potassium ATPase. So they will act on it to boost up the production of these channels or this membrane protein. And by doing so, they will increase more sodium reabsorption and more potassium secretion. So here is the basolateral side. This one will bring the sodium back to our body. This is the apical side. We will get the sodium from the urine. And by doing so, we will pump potassium into the cells. This excessive potassium will get leaking out into the urine. So we reduce the urine. So that's the aldosterone. This is where they act on. When they act on, this is what they do. All right. So that's that. Another one is ADH. ADH, okay, aldosterone secretes from the adrenal cortex. So you know that. And so they act on the distal. I think we have the picture right there. Show you that. This adrenal cortex, it secretes the aldosterone. So that's the aldosterone. ADH comes from the posterior pituitary. So here is posterior is the extension from the hypothalamus. So they are secreted from the posterior. When they secrete in the posterior, they circulating, they do several things. ADH is also called vasopressin, so they can do vasoconstriction. And here ADH. And then when they act on the kidney, they act on, where do they act on? When they act on kidney, where do they act on? They act on the collecting tubule, collecting ducts, especially in the medulla. So this channel right here, ADH will come in here to open up. You need to know that before ADH acts on it, this entire segment is water impermeable. So water cannot diffuse across. But with the ADH acts on it, it will open up water channel. Right here, right there, water opens up water channel. And then because the environment is so salty, it's so condensed, water will get reabsorbed back to our body. And this is how we regulate water reabsorption. If we need to get more water into our body, ADH coming here, causing water reabsorption. If we have too much water, then ADH will not come in here. So we basically will just pee them out. So that's the ADH. So that's that, the collecting tubule, collecting ducts. What's the difference between the tubule and ducts? Basically, this whole thing is called duct, but each duct can have multiple nephrons to connect to it. So each nephron the final portion, we call it collecting tubule. And when they all merge together, we call it collecting ducts. I think I probably has another picture like here. So this will be the collecting ducts, and this is another nephron, this is another nephron, this is another nephron. Yeah. So that's that. And that's about end. In the end, I want to going back to the, it's like looking back to the very beginning. We don't forget what is our um our purpose of this lecture or this entire course. This course is physiology. And at the very beginning, we start to have that curiosity about how does our body maintain the homeostasis. And this one, in the final one, I want to use this one to connect back to that original love of physiology. Why does our body maintain homeostasis? And I want to use this example to tell you how does our body to maintain the homeostasis of body fluid. Yeah. So our body is fluid, has a lot of fluid, but how do we maintain that? We have two ways. So in order to maintain the homeostasis, you need to have a sensor, you need to have the integrator, you need to have the effector, and response and result. Yeah. So let's take a look about the homeostasis loop for the blood fluid. So basically, the sensor will be the osmoreceptor. It's located in the hypothalamus. So in the brain area, they sense the blood thickness. Yes, blood density, basically, blood density. If the blood density is low, it means that your body has a lot of fluid. If the blood density is too condensed, is high, it means that your body lose fluid. Yeah, so that's that. Say if your body lose fluid, low water, then your osmolarity is high. This osmoreceptor sense it. So it senses your body. So here is let me draw it so you have some visual understanding. So this is your blood, right? And you have a lot of components in it. Maybe I should use a different color. Sorry about that. So this is your blood, right? So you have a lot of components in it. Ions, proteins, etc., etc. Yeah. But say, just like now, I talk a lot, talk. So this is, and I can use another one. I can do copy, maybe. Let me clear that one. Okay. Sorry, let me play a little bit. I think I can do copy. I'm not so sure. Let me double check. Can I do copy? All right, never mind. I don't think so. All right, so this is a blood. All right, that's okay. And another one. All right, so that's same thing. Your components will not lose. So you still have the same components. All right, but when before I talk, right? Our I have a lot of fluid, right? So a lot of fluid. But after I talk, my water fluid become less, right? So the fluid become more condensed. And so that condensity will trigger the osmoreceptor. All right, fair. It's a sensor receptor. It sends the osmolarity. The primary one is in the hypothalamus. Yes. And so they will tell the integrator. Integrator is the hypothalamus. but it has the extension to the posterior pituitary gland. Yeah, so that is that. And so it will release ADH. It will act on the effector. Effector is the target is the collecting tubule, collecting ducts, and basically that's the effector. And then the effect, that is a result, it will be the reabsorb water. So we reabsorb back. So we can return the homeostatic loop, return the osmolarity back to normal. So you reabsorb water back, the water back, you inject it to breathe. So we don't pee them out. So that's that. So here is ADH. So that's one pathway. This is a hormonal pathway. We also have behavior pathway because now you have less water. Just make it easy. Just drink some water. Here we go. So we have the behavior pathway. Yeah. I think I can keep the drawing. It won't be the same. All right. So same thing here. You have you lose a lot of water, and so your blood become condensed. You have the same sensor. The osmolar, the osmoreceptor will sense it. Then it's in the hypothalamus. You know it's in the brain. So it will drive you. It will tell you you have the desire to drink water. And so you will go to drink water. That is another example I told you, earlier that hypothalamus is, even though it's very small, but it releases so many different hormones to drive your behavior, to influence your behavior. You're eating, you're sleeping, your bondage action, right? Like that's oxytocin, right? Maternal behavior. Those are driven by oxytocin, by the hypothalamus. So another behavior is drinking. And so you will go to drinking. Integrator is the same thing. Hypothalamus to the cortex in the brain to give you a feeling of the thirst. Then it will drive you the action. And the effector is, go to drink. And the action of that effector will be drinking water. And then result will be you put more water into it. You return the osmolarity back to normal. So that's another way that we have multiple pathway to maintain a body's homeostasis. That's when one doesn't really work very well, we have another one. Yeah. So that's it. And that's everything. All right, let me know if you have any question, okay? Yes. All right. Bye-bye.